Um, I had talked about tight junctions, desmosomes, and then kind of hurriedly responded about hemidesmosomes before shooing you out the door last time. Is that correct? Yeah. Okay. So I described the tight junctions as like a ziplock boundary across the top of the cells that gives an impermeable barrier to the epithelium. Oh, before I dive in, what are the questions? Did anybody have any questions so far that I can answer? Everybody solid with the material so far? Good. And then I described hemidesmosomes and desmosomes I described as a spot weld, didn't I? I put the two sheets together and kind of button them together. I didn't get a chance to emphasize one of the interesting aspects of them, which is there is an element of tensegrity, which is a word that I believe was coined by R. Buckminster Fuller, if anybody is familiar with him. He's the guy that created the geodesic dome. Sometimes we talk about him in organic chemistry when we're talking about Buckminster Fullerene, carbon nanotubes, and all that stuff. Anyways, tensegrity is these tensile, in any kind of architectural structure, or these tensile cables that extend between different elements of the architecture of the structure in question, giving it an overall strength, a tensile strength, right? And so that the integrity of the structure due to the tensile strength achieved by the internal cables that hold it together. So a very simple example of this may be the spokes on a bicycle wheel, right? You can support a bicycle wheel by the axle, which is not, the spokes don't push on the axle, they're actually pulling on the axle. They're pulling on the axle, right, from the perimeter. And so these desmosomes the desmosomes and hemidesmosomes act as anchor points for these intermediate filaments, the cytoskeletal elements that give the cell its own sort of tensegrity, help it maintain the shape that it has. And also helps the cell to respond in a genuinely mechanophysical way to forces that it may feel in other distal parts of the cell. Okay. And then lastly are gap junctions. I find gap junctions to be fascinating. So they're like grommets. Now, grommet does anyone know what a grommet is? Everybody does? Nobody does? It's not the little clay character that you see in those Wallace and Gromit cartoons. It's so cute. A grommet is actually like a metal ring that you would see on the edge of a tarp or on a sail sheet for anybody that ever gets to go sailing. A grommet, which allows the passage, it connects to adjacent plasma membranes and allows the passage of various solutes to go from the inside of one cell to the other. So this creates what's called a syncytium. Syncytium, that is basically the concept that the cytosol of one cell is contiguous with the cytosol of another. So when you have an epithelial layer that is connected with gap junctions, that there is one continuous, there's a continuity of cytosol, right? And if there's an increase in some cellular component, some kind of solute in one cell, that's going to diffuse out into the others. Now, gap junctions they're not just found in epithelial cells. I'm describing them here in epithelial cells. They're found in lots of cells. In fact, you can find them in there's a high degree of gap junction expression in neurons. They're kind of an interesting structural element that I hesitate to go into deeper because it'd probably get me way off track. But yeah, they play an interesting. I'll just say they play an interesting role in the function of neuron. In the function of neurons. Yeah, leave it at that. Okay. So we can classify epithelia morphologically, and in terms of both morphologically and in terms of their layering. So there can either be simple or stratified. Now, a simple layer of epithelia is just a layer that's one cell thick, really straightforward. And then stratified is multiple strata of cells. So simple or stratified, that simple being the top row and stratified being that bottom row that you see there. They can also be categorized as squamous. The word squamous, what does that word sound like to you? Squeamish? Yeah, maybe squeamish or squished. Squished. The squamous cells are squished down. They look like a little rod there. That's not actually what they look like. They look like sort of, hexagonal or polygonal squished down blobs when you look at the top of them. And they form a tiling, a thin tiling over a surface. So squamous just means thin, actually. If somebody is squeamish, their blood is said to be thin. They're thin-blooded. They can't handle seeing a gory scene or something like that, squeamish. So squamous just means squished. Then cuboidal epithelia are, the name is pretty indicative of what you have here. They're just cubes. So a simple cuboidal epithelial layer is what you would find in the convoluted tubules of the nephrons in the kidneys, for example. And then finally are the columnar cells. And a columnar is just a long, drawn-out, a much thicker layer of cells than, for example, a squamous epithelium. And columnar cells tend to be ciliated when you find them. You can find columnar cells in the nasal passages, for example. Another category is really a histological. Another simple category is really a histological distinction. We call them pseudostratified columnar. And they say it's pseudo-stratified because, in fact, it's a simple layer of epithelial cells. But on histological cross-section, it looks stratified. And that's because the basement membrane of the cell, as it comes up, it doesn't follow a straight path. They sort of, like a bowl of spaghetti, kind of squish together. And sometimes you'll have newly forming ones that don't make it all the way to the apical surface of the other more fully developed ones. So it's a little bit of a cross breed between simple and stratified. Then on the stratified side, we have stratified squamous epithelium. An excellent example of this is your epidermis, the skin, your outer layer of skin. They can be non-keratinized or keratinized. Keratin is simply a protein, a connective tissue protein made by keratinocytes, which are the specific type of squamous epithelium found in the epidermis, and gives your skin a level of protection to the environment. Cuboidal cells and columnar cells can also be cuboidal or can also be stratified. And then finally is this another category of genuinely stratified cells that is a little bit strange. We call it transitional epithelium. It exists in a few places in the body, most importantly and significantly in the bladder. When you look at transitional epithelium, one of the characteristics of it is because of the specific morphology or the structure of the cells, it's able to stretch within the two-dimensional plane of the layer of epithelium. Why would that be a useful feature in the bladder? Absolutely. So it can stretch when it's full, and then it can come back to normal when it goes to the void volume. All right. So onto the glandular epithelia. There are endocrine glands and exocrine glands. The distinction here is that endocrine glands are going to release some sort of hormone into the interstitial fluid. So what is the interstitial fluid? What is the interstitial fluid? I should probably, the fluid between cells, yeah. There are various fluid compartments in the body. I probably should introduce that concept earlier in the course. I will talk about it at some point. But there's fluid in the blood, there's fluid in your cells, and there's interstitial fluid, right? And the interstitial fluid is just that. It's the fluid that is in between cells. So endocrine glands release the hormones into the interstitial fluid so that the hormones can then intravasate or go into the capillaries that are nearby those epithelial cells and then be carried around by the bloodstream. So that's endocrine glands. On the other hand, and I'm not going to talk about them much until we get to the endocrine chapter, but exocrine glands are going to produce secretions of some sort into ducts that are present on the epithelial surface. So this happens through a number of different methods: Merocrine, Epicrine, and Holocrine secretion. We'll go through each of those in a little bit of detail in the next slide. But the point here is that these exocrine glands are going to release their cellular contents into some sort of ductwork that is not available to the bloodstream. So whatever secretion the exocrine gland is making should not be getting into the blood, right? It's being released onto the surface of the cell, the apical surface of the cell, which in conjunction with other similar cells forms some kind of a duct that is doing who knows what. There's all kinds of exocrine glands. We'll talk about a few of them. I used to go through the microstructure of the glands, but I find it to be not very useful, actually, so I'm not going to do that. But I will talk about the different types of exocrine secretion. And the three of these, Merocrine, Epicrine, and Holocrine, form a kind of a continuum. The first is Merocrine. So Merocrine glands, the way a secretion works in general is there is some sort of gene product that goes to the endoplasmic reticulum to get translated by the ribosomes, and then it moves on to the Golgi apparatus for post-translational modification. Hopefully these words are not new to you. And once it's in the Golgi and it's finally finished being made, it gets packed up into a little vesicle, a little balloon of lipid membrane, lipid bilayer, that is going to move towards the surface of the cell, and that membrane is going to fuse with the apical membrane, the plasma membrane at the apical surface, and release what was inside the little vacuole, the little vesicle, onto the surface of the cell into the lumen of whatever duct we're talking about. So this involves exocytosis, probably involving calcium. So some examples of this are your sweat glands. And there's a couple different types of sweat glands. I'm probably not going to talk about them because of time, but there is the type of sweat that your body makes, the watery sweat that your body makes for evaporative cooling, right? There's a couple different types of sweat. There's also sort of oily sweat that comes on after pubescence that is more odoriferous, smells a little bit stronger, has the pheromones in it that we all either like or dislike so much. The next type is apocrine secretion. And the secretory product is still made in the Golgi apparatus, but rather than exocytosis, there is what's called cytoplasmic shedding. So on the maybe distal or the apical third of the cell, along that tight junction, it sort of pinches off the top half of the cell, top third of the cell, and releases that whole chunk of the apical surface into the lumen of the duct, and then it begins to break down more and more. So the prime example, there's only a few examples of this, but the prime example is the mammary gland. It's also in the tarsal glands of the eyelid, or the ciliary glands of the eyelid. Why would the mammary gland use apocrine secretion instead of merocrine secretion? What do you guys think? What is the mammary gland doing? Let's start with that. What's the mammary gland doing? Pretty straightforward. You should all get that one. It's making milk. And what's in milk? What is milk? Well, definitely. It's got calcium. What else is in milk? It's not just pure calcium, though. There's other stuff in there. What's that? It's not a trick question. I'm really asking an easy question here. So, yeah. There's proteins. Absolutely. Is that all that milk is? There is certainly proteins. We know there's proteins in it because if you ever try to make paneer or any kind of uncultured cheese, anyone ever done that before? You scald the milk just a little bit and you put some lemon juice in and it all curdles, the curds and whey. That's a lot of the protein congealing. What else is in milk? What's that? Lactose. Yeah, lactose is one of many sugars found in milk. Yeah, there's carbohydrates. So there's calcium. There's micronutrients. We'll just say, we'll make a big category: micronutrients, proteins, carbohydrates. All that stuff's reasonable. What else is in milk? What's that? Water, absolutely. Fat, thank you. That's what I was looking for. Fat. What's another name for fat? Lipids. What do lipids do? What do lipids do? I know, I'm not expecting like a huge discourse on this, but what's the function of lipids? Okay, before I do that, tell me something that's unique about lipids when compared to the other things that you listed. You listed calcium, you listed proteins, you listed carbohydrates. Just trying to get you to think about this, why this makes sense, the structure-function paradigm. What about proteins, carbohydrates, calcium, and the thing, I don't know, maybe it was you, said water? They're all soluble in water, aren't they? They're all soluble in water. What about fats? Are they easily soluble in water? No. How do we get the fats into the milk? How does that happen? Well, where in your cell, in a typical cell, not an adipocyte, which is a fat vacuole, but in a typical cell, where are you going to find the lipids? In the membrane. In the membrane. In the membrane. So, therefore, it might make sense to release a whole bunch of membrane into the milk, okay, and then to have that break down into little vacuoles and little micelles of lipid bilayer. So milk is actually a suspension. It's a colloidal suspension of little vacuoles of fat. This is how it happens: is by apocrine secretion. the apical end of these epithelial cells shed, and this is how we get both water-soluble nutrients and fat-soluble contents, lipid, get dispersed into the fluid. This is why we call it apocrine secretion, because the apical portion of the cell sheds off. All right, and then holocrine secretion. This is super radical. Oops, let me get that for you. Yeah. Holocrine secretion, these cells just blow up. They just blow themselves up. And because of that, holocrine secretion glands need to be stratified. Because if there's not lower levels or more germinative levels of the holocrine epithelium, then you're not, which are constantly dividing and replacing the ones that are breaking apart on the surface, then you're in trouble. So an example here is the sebaceous gland. Sebaceous glands are essentially the glands that give you pimples. They're oil glands on the skin. They produce sebum, skin oil. So what's the purpose of a sebaceous gland in general? I just said to make skin oil. What is it really trying to do? Yeah, it's trying to stop water from getting in, like a water permeable barrier. It's also trying to protect your skin from other pathogens that may be getting in. It's trying to create some sort of a barrier that is going to prevent injury or attack by some kind of pathogen. So it sort of makes sense that the cell, if it blows itself apart, maybe it fills itself up with whatever kind of oily secretion it's going to make, and whatever cellular contents that it has, it's going to blow itself apart, get released, and all that cellular smudge will form a kind of protective barrier along the outer edge of the epithelium. All right. So holocrine secretion. Each of these structures, each of these forms should correspond in your mind to the function that it needs to serve. So, and the kinds of questions, those leading questions that I asked there, that's the kind of critical thinking thing. You know, a lot of students often ask me what kind of questions I ask on the midterm or the final. And when I say critical thinking, what does that mean? It means that I give you some information and given what you know, try to get you to think deeply about what it may be happening in a situation. You see what I'm saying? That's different than regurgitation of information. Okay, so that's all I'm going to say about epithelium. And I'm going to move on to connective tissue. So I have trimmed the connective tissue unit pretty significantly. I'm going to talk about some of this stuff here, most of this in fact, but only as it's necessary to support what I want to talk about in other units. So what does connective tissue do? Well, it connects things. It binds organs together in one way or another. For example, as tendons connecting muscle to bone, or ligaments connecting bone to bone. Connective tissue, there is the supportive connective tissue of bone and cartilage. It's a type unto its own. And bones provide physical protection in terms of the cranium, the ribs, sternum, etc. And immune protection. So there's the fluid connective tissue, the blood cells, the red and the white blood cells, particularly the white, obviously produce various factors that are going to constitute your immune system. So antibodies and different inflammatory hormones. And then the cells themselves can be phagocytic, meaning that they can attack or cytotoxic. They can attack in one way or another whatever is invading the body. Or maybe it's a rogue cell in the body, a cancer cell in the body that is a cell, but that has gone off the tracks. Movement. So the bones provide the lever system that the muscles work on, and adipocytes act as storage vacuoles for the fat. Calcium can get stored in bone. Phosphorus gets stored in bone. Heat production. So babies are fun to talk about. And adipocytes, maybe if I keep clipping along here, we'll get to see a picture of my baby daughter when she was a fat little baby, and talk about brown fat. And then the transport in terms of the erythrocytes, the blood. And then lastly, it just serves as the basal lamina for all epithelium in the body. So there's any epithelium that is somewhere that is connected to a basal lamina, that is being constructed by a connective tissue. And it essentially just forms the fascial planes, the fascia that's in your body, the layers of connective tissue that separates and defines the boundaries of various organs. All right. So we're going to walk through all this connective tissue. First, the connective tissue proper, and then I'll touch a little bit on the supportive and fluid connective tissues. Here is the basic breakdown. There's 9 types of cell populations that you'll see in connective tissue. Fibroblasts, cytes, adipocytes, mesenchymal cells, macrophages, lymphocytes, mast cells, microphages, and melanocytes. You don't need to scramble to scribble it all down. I'm going to walk through them one at a time here. Connective tissue itself is broken into this hierarchy you see in the top. There's the proper connective tissue, which is distinguished as loose or dense, and then there's these specialized forms: the supportive connective tissue in the bone and the cartilage, and then the transport connective tissue in the blood. All right, so let's start looking at the cell types. First, there are the fibroblasts, which are not terminally differentiated cells, much like an osteoblast becomes an osteocyte. Fibroblasts are able to differentiate into fibrocytes. The fibroblast is by far the most common connective tissue cell in the body. It's in any kind of connective tissue proper that you would find anywhere in the body. And it produces fibrin. It also secretes hyaluronic acid. Hyaluronic acid is a glycoprotein. You remember? Seems like you're remembering hyaluronic acid fondly. Yes, absolutely. Yeah, it's an acidic or anionic one. The conjugate base is an anionically charged glycoprotein. It's quite slippery. It acts as a kind of a cellular cement, is the word I use there. Fibrocytes are also in all connective tissue proper, and they are more maintenance cells. All right, adipocytes. Fat cells, not especially exciting under the microscope, although kind of beautiful, I guess. They're mostly just this white, hypodense staining cell where the cytoplasm is completely taken up by a single vacuole that's full of lipids, and all the cellular contents are slammed to the side. Adipocytes store fat, but they also act as endocrine cells in the production of leptin, which we'll talk about in the GI tract. Mesenchymal cells are stem cells that get mobilized when you have some kind of an injury or there's an infection, there's some tissue that needs to be repaired. And so a mesenchyme is a germinative cell. meaning it's a type of stem cell. It's not terminally differentiated. A mesenchymal cell can go in multiple directions. It can go in multiple directions. It can become a fibroblast, which will then become a fibrocyte. It can go towards macrophages. A mesenchymal cell is kind of like a grandparent to a number of connective tissue lines. Macrophages, they're amazing things. You guys can go on YouTube sometime and look for videos, electron micrograph videos of macrophages doing their blob that ate Philadelphia thing. It's pretty incredible to watch. There are these cells that send out these pseudopodia and these long projections and find pathogens and then slowly surround them and absorb them. It's kind of terrifying if they weren't on your team, and so tiny. So there's a couple different types. There's macrophages that are anchored in a single place. They're not going to go anywhere. They're just waiting for something to come along to them. And then there are the freewheels that are able to migrate around the body in search of pathogens. So here is a macrophage tearing after a whole host of these little bacteria doing this is Mortal Kombat right here. All right, mast cells. So these, I gave you these 9 cell types. This is all super general, and there are a lot of gradations and subcategories of each of these. Mast cells are a kind of pro-inflammatory cell that are going to release various inflammatory cues like histamine or heparin in response to an injury or infection or what have you. Mast cells are not basophils. Basophils are a type of a leukocyte or white blood cell that also contain histamine and heparin. For a long time, people thought basophils were mast cells, but they understand now that that is not correct, and there are some subtle differences. They look really similar, and it's hard to tell the difference histologically between the two of them, and functionally there's not a whole lot of difference. All right, lymphocyte. So lymphocyte is a very general term. It just means white blood cell that is going to make an antibody. And a lymphocyte can be a T cell, a B cell. Within the T cell and B cell category, there's tons of different subtypes, and you'll have to take an immunology class to begin to start untangling all that. There's some pictures of them, at least. Maybe I've whet your appetite for immunology. Microphages are another type of phagocytic blood cell. The microphages are the granular leukocytes. The lymphocytes are the agranular leukocytes. I didn't make that distinction in the last slide, but the granular leukocytes are eosinophils, neutrophils, basophils. I had mentioned basophil when I talked about mast cells. And these are a category of cells that I talk about in my anatomy class, but I'm not going to talk about much here. Melanocytes are a type of cell that produces melanin. It's a UV absorbing pigment that gives your skin its characteristic color, and they produce and store the melanin and are able to send out the pseudopodia into the skin that then release these vacuoles that will invade other keratinocytes in your skin and give them melanin to help protect them from the deleterious effects of UV. So people with darker skin, there is a misconception that people with darker skin have more melanocytes. This is incorrect. In fact, people with darker skin just have melanocytes that produce more melanin. Everybody has approximately the same density of melanocytes. It's just people with darker skin produce more melanin and thus have more protective immunity against the effects of UV radiation. I'm probably not going to talk about skin cancer at all, but melanocytes are pretty interesting in how that plays out. So here are some different types of fibers that this cast of characters can make. The three big ones are collagen fibers, which I guess the word I use to describe collagen is the Hauser rope, like those giant ropes that they use to hold big tanker ships and what have you into dock. Those thick, strong cables strung along the axis of the fiber, not very elastic. So if you pull a collagen fiber, it's going to resist force. Whereas on the other hand, elastic fibers, if you pull an elastic fiber along its length, it will not resist the force. It will stretch, still maintaining its structural integrity, but then when you release that force, it'll come back to its original shape. They tend to be branched, wavy, elastic fibers. Then finally are reticular fibers. Maybe I should put that list in a different order. Reticular fibers are going to, they kind of look like, let me think of a, I don't know a good macroscopic analogy for it. I'd have to think about it harder. But reticular fibers are ones that have projections out in multiple directions and form a strong three-dimensional support network. So you often find reticular fibers in different capsules to organs. So the capsule around the kidney, for example, it's kind of a tough coating on the outside of a kidney. That is made of reticular fibers. Reticular fibers just resist force in three dimensions. So you're going to find this also in like dense irregular connective tissue, which is what is in the dermis, the reticular layer of your dermis. We'll see. We'll talk about that when I get to the skin a little bit. All right. Lastly, here is ground substance. So I'm just giving you the cast of characters so that we can talk about the types of connective tissues that we see. In ground substance, I talked about the osteoid in the bone, didn't I? That forms the matrix of the bone. The osteoid is that sort of proto-collagenous material. It's not collagen, it's osteoid, but it's similar to collagen. That's made by the osteoblasts that then gets calcified by the osteocytes. Ground substance is kind of like that. It's this goo. It's this goo that your connective tissue cells like to live in. Right? It's a high viscosity, colorless, clear goo. And it is invested with the collagen fibers. It's invested with elastic fibers. There's going to be the panoply of cell types that I very quickly flashed through there, found in the ground substance. It's going to have a high viscosity because it is full of an array of different macromolecules, and they fall into three general categories: glycosaminoglycans, or GAGs, proteoglycans, and various adhesive glycoproteins. Have they talked about that in? They don't talk about that stuff in biology, right? You would have to talk. Biochemistry would give you that. Has anyone taken biochemistry here? Just a couple. Yeah. Right. Well, anyways, glycosaminoglycans, proteoglycans, and adhesive glycoproteins. There are 3 letters that you'll notice are the same. A 3-letter combination that is the same in all 3 of those words, and it's GLY. GLY. In fact, GLYC. 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 What does that word combination indicate? What does that refer to? Glyc. Anyone? No? It refers to sugars, carbohydrates. Like, for example, your glycemic index of a food is how rapidly it will increase your blood sugar level. Yeah, so let's look at these a little bit. Glycosaminoglycans are these carbohydrate repeats. These carbohydrate repeats. So a carbohydrate in general is simply a carbon backbone chain, usually 5 or 6 carbons in length, that is polyhydroxylated, has a lot of OH groups all over it, and forms these long chains. And then they adhere to these protein cores. There's a protein core in the cartoon here depicted in purple. And the actual carbohydrate portion of it can be chondroitin sulfate or high hyaluronic acid, hyaluronate or urinate that I talked about earlier. If you look at these structures, I put the structures up there not so that you can memorize them or to freak you out at all, but simply that you can see on them there is, in addition to all these hydroxyl groups, these OH groups, these alcohol groups on the sugars, there is a significant amount of anionic charge, negative charge. You see all those, the conjugate base of the carboxylic acids there, the COO minus, and then we see the sulfate group on what's called the hydroxymethyl tail of those sugars. Those anionic charges, what do we know about like charges? They repel one another. Yeah. And so because of that, that's going to help the chondroitin sulfate, these glycosaminoglycans, to fluff out, to want to expand as much as they possibly can to keep the negative charge away from each of the other ones. And it's going to make it pretty slippery. On top of that, it's going to want to be hydrated. These are all really polar molecules. All those hydroxyl groups want a hydrogen bond with water. All those negative charges are going to want to be hydrated. And this is what's going to help to make the ground substance of connective tissue really viscous. Why might it be a good idea to have a really viscous substrate in your connective tissue, which is underlying your epithelium? Exactly. So say you scratch your skin or whatever, and a bacteria gets in there. You don't want that bacteria to just be running rampant. You want it to be running through a pool full of Jell-O, right? And that's kind of what it's like. It's like running through a pool full of Jell-O or honey or something like that, if you can imagine what that would be like. Hard enough to run through a pool, but much harder to run through Jell-O. So I don't want to say anything else. We're going to come across some of these characters again when we talk about joints and the vitreous of the eyeball. We get to talk about that. All right, I wish we could stay here longer, but we can't. So, different types of connective tissue. Let's skip dense, regular connective tissue. These are just tendons, I guess. I don't need to talk about that much. It's just densely packed in a regular array, resists force along the axis of the array. It's just tendons. Now, dense irregular connective tissue is packed in this kind of crazy three-dimensional pattern. This forms the the reticular layer of the dermis, the deepest layer of the dermis, not the hypodermis. There's epidermis, dermis, and hypodermis. We'll see that when we get to the cutaneous membrane next time, I guess. But this dense, irregular connective tissue forms a three-dimensional lattice that resists force from any direction. And elastic tissue is found in the body in different places. For example, between the ligaments in the spine. And this is what enables your spine to be able to bend in all those directions and then come back to its original shape. Very stretchy. Okay, that's all the categories of dense connective tissue. To the loose connective tissue. One of the most important categories is areolar tissue. Now, areolar tissue is all over the place in the body. It's all over the place. You're going to find it beneath virtually all epithelia, so it's forming the basement membrane or the basal lamina of like all epithelium. It's going to be found below as the connective tissue layer and all the serous membranes. We'll talk about what that means next time. You're going to find it forming the epimysium of muscles, the connective tissue sheath of muscles. And one of the things it does is it provides an excellent substrate for the passage of capillaries. So you have to have capillaries to get close to the epithelium. Why can't the capillaries just move through the epithelium? You guys remember that one? What did I say? It's avascular. That's it, Zoli. Yeah. It's avascular. And so there needs to be some kind of sort of service conduit, I guess, for lack of a better word. That's the areolar tissue, this areolar connective tissue. It's just ideal material for little capillaries to send out their rootlets and supply the skin. So, for example, if you scratch your skin gently and you pull away the epithelium, it won't bleed. But if you scratch it more deeply and you get down into the connective tissue of the dermis, the papillary layer of the dermis is areolar tissue, and that's when it'll begin to bleed. You know you've hit dermis when a scratch bleeds. All right, reticular tissue, not too much to say here beyond what I said about reticular fibers. Reticular tissue is made up of reticular fibers, forms the capsule, or the word is stroma, actually, for various lymphatic organs, other organs. That just means the body of the organ, not just the capsule, but the body of it. All right, I don't want to get hung up on that. So this is the last slide I'll talk about, then I'll let you out. Actually, I'm not going to do it. It's time now, according to that clock. I'll let you out. We'll talk about adipose tissue and my daughter next time. She's 6 now. I don't know. Any questions?